This is a GCSE presentation looking at health issues. To get started, there are a number of different diseases listed in the middle column, and all you have to do is determine if each one is a communicable disease or a non-communicable disease. Remember, communicable diseases are ones that can be transferred between people, so they're contagious and they're caused by a pathogen. Whereas a non-communicable disease cannot be passed on, it is not contagious, and it is not caused by a pathogen. Pause the presentation now, work out which disease goes to which side, and when you're ready you can restart and check your answers. Pause now. So for each of the diseases, measles is a communicable disease. It can be passed on, it is contagious, and it is caused by a pathogen. Cancer is non-communicable. Coronary heart disease is non-communicable. Salmonella is communicable, it can be passed on. Diabetes is non-communicable, and malaria is communicable because again it can be passed on from person to person. In this lesson we're going to look at some health issues. We're going to look at factors that impact on health, and we're going to look at the concepts of sampling and some ways that data can be presented. Health is a state of physical and mental well-being, and there's a number of different variables that can have an impact on it. On the right hand side you'll see a list. You need to be aware of all these things can have an impact on a person's health, both physical and mental well-being. It's also possible for diseases to interact, so that one disease then makes you more likely to suffer from something else. For example, some viruses increase your risk of cancer. Some certain types of pathogens and viruses and infections will actually compromise your immune system, and as a consequence you're more likely to pick up other infections. Poor physical health is often connected to poor mental health, and often poor physical health over a long period of time can lead to things like depression and other disorders. In the exam, often in this topic they'll give you questions based around frequency tables, histograms, scattergrams, as well as the traditional line graph and bar chart. Remember, a frequency table is a way of arranging large amounts of data into a kind of summary table. If you look on the right hand side, we've got a whole bunch of raw data, 25 data points, and we've literally grouped them into 3 groupings. And by putting them into 3 groupings, 0 to 10, 11 to 20, and 21 to 30, we've then taken a tally. Remember, 1, 2, 3, 4 up and down, and 1 horizontal to get tally marks. And then we've counted the frequency. And when we look at the raw data, we have 5 inside the group 0 to 10, 8 inside the group 11 to 20, and 12 inside the group 21 to 30. If we wanted to, we could present that as a table, and what we would do is we would present the x-axis in the same way we do a bar graph, but instead of doing bars we would simply put x's for the frequency. We can also have what are called histograms. Histograms are a bit like bar charts, but unlike bar charts, all the bars are not the same width. In a bar chart, the area that a particular bar takes up represents the frequency of that particular variable. And you have to get the combination of how many along the x-axis your particular grouping takes and how high that particular bar has to be. And there's a method of working it out. If you look at the data in the top right hand corner, you can see that the first grouping we've got is between 0 and 3. That has 4 points, 0, 1, 2, 3. So the number of points in the group is 4. In the second group we have the values between 4 and 8, and that gives us 5 points. We've got number 4, 5, 6, 7, and 8. And in the last grouping we have 2 points, which are 9 and 10. The frequency is just simply how many people, characteristics, whatever it is you happen to measure, fall into each group. So for the first group 0 to 3 we had 8, for 4 to 8 we had 15, and for 9 to 10 we had 8. people, for example. So to work out the frequency density, we simply work out the number of things that fall into that category, which is the frequency, and we divide it by how many points on the x-axis are included in that category, which is the number of points in the group. And if we do that, the frequency densities come up as 2, 3, and 4. So when it comes to doing a histogram, we would have the first bar on the histogram has to go along the x-axis from 0 to 3 because that's the group. And its frequency density is 2, so it goes to a height of 2. The second grouping has between the numbers 4 and 8, so we continue the bar, there's no gaps. We continue the bar all the way across up to 8. But the height this time of the frequency density, it has to go up to 3. And then on the last one, we have only got the groups all the way up to 10. So again, we continue from where the last group ended up to 10, and we make that up to a height of 4. Scatter diagrams are essentially a type of line graph where we have one variable on the x-axis, one variable on the y-axis. And what we're looking for is a relationship between those two variables. And we know there's a relationship the more we see a clear pattern in our plots and data. If you look at the right hand side, you can see 3 different graphs. The top graph shows a very clear positive correlation. By a positive correlation, we mean as one variable increases, the other one increases. But you can also see that all the x's for the plots fall exactly on the line. In the middle graph, we can see that we have still got a pattern, it's just not as good as the first graph's pattern. This time around, however, we have what is called a negative correlation, because as we increase one of the variables, the other variable decreases. And in the third graph, we have what's called no correlation. There is no clear pattern in those set of results. There is no line of best fit. So there is no relationship between those two variables. Now, sampling is simply when we take a small proportion of a population so that we can carry out some research without having to deal with every single organism in the entire population. But it is important that when we do take a sample, that the sample has to represent the original population. So for example, if I was to take a sample from a bunch of students, I would have to take some male, some female, and I'd have to take some from every year group. If I only took my sample from year 7, well, that would be a biased sample because the sample does not represent the entire school population. Remember, samples should be taken at random. We shouldn't have any pattern or bias in what we're selecting, but we can take regular intervals. So for example, I can randomly pick 10 places around the school to ask students questions, but I'm going to ask every 5th student that walks through the door. I'm taking regular intervals. Just one thing to be aware of, one of the classic questions that you can get is about using a quadrat, which is what's in the picture. And we use that and we put it on the ground, and what we do is we can count the number of plants inside it, and that's a way of sampling. And one of the classic things is that if we throw that quadrat into a field, is it an example of random sampling? If we close our eyes and throw it, is that random? Well, believe it or not, the answer is no, because all parts of the field are not equally likely to be included. The place that you're standing on is not going to be included. The places behind you are not going to be included. And you're only going to be able to throw that quadrat so far, so beyond that point, you're going to have no chance of picking up any information. So it is not random. So you just got to be very careful what you think is random and what is actually random. Actually random means every single thing in that particular population has an equal chance of being picked in a sample. Now we've got a number of risk factors and we've got some diseases. what I'd like you to do is pause the presentation and match the risk factors on the left-hand side with the disease or problem they're most likely associated with on the right. So pause the presentation now, and when you're ready, you can restart and check your answers. Well, to get started, diet, exercise, and smoking is actually linked to the cardiovascular diseases. Obesity is linked to type 2 diabetes. Remember, type 1 you're normally born with, type 2 is the one you can develop. Alcohol intake is the liver and brain damage, and it slows your reaction time down. That's why drinking alcohol and doing things like driving and operating heavy machinery is a major risk, because your reaction speed is much slower. Smoking, of course, is linked to lung disease and lung cancer. Carcinogens, well, carcinogens is the group name given to a bunch of things that will all cause cancer. And lastly, alcohol and smoking will affect unborn babies. Now, we mentioned cancer before. You do need to know a little bit more about cancer. You need to know what cancer actually refers to. Well, cancer is what we call a cell that is growing and dividing uncontrollably. When we get a group of cells, we call that a tumor. There's a number of things that can cause cancer, including viruses infecting certain cells. Sometimes it can be down to carcinogens, such as UV light. What we can also have is some level of genetically inherited factor. It doesn't mean that you will definitely get it. It just means some people have a high risk and some people have a low risk. And if you're high risk, it's much better for you to take certain steps to reduce your likelihood. So for example, if you're a high risk family, you wouldn't want to smoke. You maybe even decide not to take alcohol to reduce your chances of getting cancer. Even if you're low risk for getting cancer, things like not smoking are going to make it even less unlikely for you to get cancer. Now, when we talk about cancer tumors, there are two groups, and you need to be able to talk about both groups. We have the benign tumors and we have the malignant tumors. And you have to be able to describe some of the characteristics of a benign tumor. So a benign tumor grows slowly. It will not spread to other tissues or membranes inside a body. It's located in one area and it's very easy to remove with surgery. Malignant tumors, however, grow very rapidly. They can spread around the body and form what we call secondary tumors, which is a process known as metastasis, and they are difficult to remove and often involve radiotherapy and chemotherapy. So make sure you know the two types of tumors and make sure you know the characteristics of each one. There is a cost with non-communicable diseases. And the cost, when we think of cost, immediately everyone thinks finances, but there's not just a financial cost. There could be serious impact on family members' health and well-being. A high level of abuse of certain substances like alcohol and cigarettes can actually place strain on very limited local and national health services. And of course, we do have the financial cost of treating diseases, particularly as many of these diseases can be avoided with a healthy diet and regular exercise and good lifestyle choices. Just to finish off, write down as many terms as you can from the lesson, spread them out over a page, and then try and build a web connecting as many of those words as possible. Even better is if on each of the lines between the words you explain how those terms are connected.